a disproportionate share of the genetic differences between humans and chimps are genes that code for transcription factors. And that makes perfect sense. You get a change in some gene coding for some structural protein, and maybe your muscles will like bend a little bit this way instead of that way, or who knows what. You get a change in the gene coding for a transcription factor, and you will have invented all sorts of novel networks. So a disproportionate share of what has gone on in evolution differentiating us from chimps are changes in transcription factors. This was a big triumph, big support for a view that came out in the 80s. There were parascientists at Berkeley, King and Wilson, and this iconic set of studies they did. They were the first people to come up with the 98% business there, and using very primitive molecular techniques that have since been affirmed. And they also came up with a prediction, a purely theoretical one, which is the most interesting changes that will occur in evolution are in regulatory parts of DNA rather than in the coding parts for protein. And everything since then has supported that, including things like you want to turn an ancestor of a human and a chimp into something that will look a whole lot different from a chimp, turn into us, change stuff with regulation, transcription factors, promoters, etc. What you see with that is this is endlessly networks for amplifying effects for macro changes. And there's a new person in the department, bio department, Hunter Fraser, a new assistant professor who works in this area, showing just how much evolutionary change is being driven by changes in regulatory parts of the DNA world rather than the genes itself. Changing a little bit about how this protein works, that's okay. Changing if-then clauses for entire networks, that's hugely important. Another interesting factoid, there's now been the genomes of, about 100 different species sequenced, and ranging from really short genomes with, how many genes in there, to the longest ones. When you pile them up and you look at them as a function of how long their genomes are, the more genes you find in a species, the greater the percentage of those genes are that are transcription factors. And this makes wonderful sense as well. You got one gene, and you only need one transcription factor. You got two genes, and you could milk maximal information out of them with 3 transcription factors. You transcribe A, or B, or AB. You've got 3 genes, and there's 7, and the equation is 2 to the nth minus 1, or whatever. But what you see is, by 4 genes, you're up to 15. By 5 genes, you're up to 30. You're having an exponential, a dramatic increase in the number of transcription factors you need to take advantage of all the possible combinations of networks of gene expression. The larger the number of genes you find in an organism, the greater the percentage it is of transcription factors. In other words, get tiny little micro changes in DNA coding for transcription factors, splicing enzymes, promoters, and you're going to have big major consequences.